In this part, I want to talk about the active process. The active process or active transport is a type of membrane transport which need energy. And we can divide it into active transport and vesicular transport. The active transport is movement of solute against its concentration gradient. It means that we need to use energy to send substances from the place where their concentration is lower to the place where the concentration is higher. And this active transport can maintain the gradient between cells and interstitial fluid. And the source of energy determines whether movement is primary or secondary. First, I want to talk about the primary active transport. In the primary active transport, we use the energy directly from breakdown of ATP. You know that the ATP is a source of energy which is made in the mitochondria, and then this ATP can be used for primary active transport. For this type of transport, we need a process which we call it phosphorylation. The phosphorylation means that one ATP molecule is broken, as you see it here. You see that when we break down one ATP, we can have one ADP or adenosine diphosphate and one phosphate group. This phosphate group can attach to transport protein. This process is called phosphorylation. By this phosphorylation, we have a change in the shape of protein, and then we have movement across the membrane. In this picture, I want to show you one type of active transport. We have one ion pump in the plasma membrane of red blood cells. This pump, as you see here, is called calcium pump. The function of calcium pump is sending calcium ion against its concentration gradient from inside to outside. Inside of the erythrocyte or red blood cell, the concentration of calcium ion is lower than outside, but our cell use energy to send more calcium ion out. For doing this transport, first the ATP is broken. As you see here, we have ADP and phosphate group. This phosphate attach to our pump here, which is a transmembrane protein, and the shape of this protein change, and the calcium can go out of the cell. We need to send calcium out because we want to decrease the rigidity of our erythrocyte. The calcium ion accumulation in each cell in our body can make rigidity and stiffness, and we don't want it for our erythrocyte because our erythrocyte should be flexible enough to pass through tiny capillaries. So one of the pumps which we have in our body is the calcium pump, which we have it in the cell membrane of our erythrocyte. The other types of transport, primary active transport, is called sodium potassium ATPase pump. If you look at this picture, you can understand how this pump works. We have first phosphorylation of the ATP. When the ATP is broken, we have ADP and phosphate group. And this ATP energy is used for sending 3 sodium ion out of the cell and 2 potassium ion inside the cell. Although we have more sodium ion outside and more potassium ion inside. So as you see here, the ATP is broken down and we have ADP and phosphate group. When the phosphate is attached to our pump, the shape of our pump change, and you see that this way become open and 3 sodium ion go out of our cell. At that time, as you see here, the location, the site for attachment of potassium become open. The potassium attached to the pump, again the shape of our pump change and potassium release inside the cell. And one phosphate group which is attached to our pump again released in our cells. So you see that we use one ATP and we send 3 sodium ion outside and 2 potassium ion inside the cell. The other way for transport is called secondary active transport. The secondary active transport is also called co-transport or cold transport. This is the movement of substance down its concentration gradient. Put another way, the kinetic energy of the substance moving down its concentration gradient across the cell membrane can provide the power to pump the other substance against its concentration gradient. The sodium gradient is often used as the source of energy because its concentration gradient, the plasma membrane, is extremely steep. We have about 99% of sodium ion in the interstitial fluid with only 1% inside the cell. The sodium concentration gradient has potential energy that is this is the potential energy which is converted to kinetic energy when sodium ion move into the cell down its concentration gradient. We have two types of secondary transport. One type is called symport and the other type is called antiport. The meaning of symport is this: when two particles move in the same direction. And the meaning of antiport is when two particles move in the opposite direction. In the transport, look at here. First, this sodium ion, which is more outside, do diffusion, and the energy which is provided by moving this sodium ion inside the cell is used for glucose to come to the cell against its concentration gradient. And in the other picture, you can see that here we have sodium ion and here we have hydrogen ion. The sodium ion move inside the cell down its gradient, and this energy is used for movement of hydrogen ion go out of the cell against its concentration gradient. This is the meaning of secondary active transport. And the other type of active transport is called vesicular transport or bulk transport, and it involves energy input to transport large particles across the cell membrane by a vesicle. The vesicles are membrane-bound sac which is filled with the material, and the vesicular transport has two different types. One type is called exocytosis and the other one is called endocytosis. The meaning of exocytosis is sending large particles out of the cell. When our cell makes some macromolecules, they are too large to pass across the cell membrane. So, for the passage of these particles, we have a specific type of transport which we call it exocytosis. At the time of exocytosis, the vesicle which contain our molecule which need to go out come and near the plasma membrane. Then it attached to the cell membrane, and you need to know that this attachment need ATP. So when we have this attachment, one ATP is broken and we have ADP and phosphate. So the vesicular transport is a type of active transport because we use energy. Then, as you see in this picture, a pore become open and the particle go out of the cell. This is the meaning of exocytosis. Release of material, release of large macromolecule out of our cell. And when we have this release, as you see it in this picture, the length of this vesicle wall added to the cell membrane. So at the time of exocytosis, the length of our cell membrane increase. The other process which is opposite to exocytosis is called endocytosis. Here you can see endocytosis. We can divide in endocytosis to two different groups. One group of endocytosis is called cell drinking or pinocytosis. One type of endocytosis is called cell eating or phagocytosis. And now I want to show you these two different pathways. First, for the pinocytosis or cell drinking, when some fluid wants to come to our cell, as you see in this picture, they come near the cell membrane, they make one invagination or one depression in the cell membrane, and after that the two sides of this vesicle attach together, and then the vesicle pinch from the cell membrane and come to the cell. We call it pinocytosis or cell drinking. When it wants to detach, we need to use energy. And the other type of endocytosis is called phagocytosis, when our cell wants to eat something. As you see here, this is a particle, and most of the times phagocytosis take place when our white blood cells want to engulf pathogens and destroy them. This particle, maybe this is a nutrient, maybe this is a virus, bacteria, fungi, or parasites, come near the cell, and our cell can make pseudopodia or false feet around this particle. And then these two sides are attached together and the vesicle release inside the cell. When the vesicle release inside the cell, we need to use energy, and we call it phagocytosis. We have the other type of endocytosis, which we call it receptor mediated endocytosis. It means that endocytosis cannot take place in all parts of our cytoplasmic membrane. We need to do endocytosis in a specific location. And this location are some pits, as you see here, which are coated with a protein, and this protein is called clattering. We call this pit clattering coated pit. Then the particle and its receptor attach together, they move across the cell membrane until they reach the pit. When they come to the pit, then these two sides are connected together and the vesicle releases in the cell. This type of endocytosis is called receptor-mediated endocytosis, and it can enable the cell to obtain bulk quantities of a substance. The transport of cholesterol from the blood to cell takes place by this method. The cholesterol is bound to a low-density protein in our blood, which we call it LDL. And then these LDL can attach to the plasma membrane. It moves on the surface of plasma membrane until it reaches this pit, the clattering coat pit, and then it can come inside the cell. And when we have hypercholesterolemia, it means that we have some defect in accepting these cholesterol in this clattering coated pit. Sometimes we don't have the receptor, sometimes we don't have enough clattering here to bring this cholesterol inside the cell. So this is a genetic problem which we may have. And when the cholesterol remain in our bloodstream, it can make atherosclerosis in the blood vessels. It can deposit inside the blood vessel and make our blood vessel become narrow, and it can increase the risk of heart disease and heart problem. And now you can answer these questions. How does oxygen diffuse into a cell and carbon dioxide diffuse out of the cell? You can compare and contrast how an ion like sodium ion is transported across the plasma membrane versus how a polar molecule can move. And you can talk about osmosis. You can now answer the questions of tonicity. When we put a cell in the isotonic, hypotonic, or hypertonic solution, what happened to our cells? And then what is the meaning of active transport? How can we divide this active transport to primary and secondary active transport? What is the meaning of vesicular transport? In which type of vesicular transport we can increase the length of our cell membrane, and in which type we can decrease the length of the cell membrane. In the next video, I will talk about the membrane junctions and the organelles which we have in each cell.